All right. Well, after giving our previous colloquium, I have the much greater pleasure of hosting Amanda Moerhing today. Professor Moerhing is a leader in the study of speciation, both in terms of its genetic architecture and its molecular mechanisms. She came originally as from an undergraduate background at Pacific University. She did a PhD at North Carolina State with Trudy McKay. She did postdoctoral work at Duke with Mohammed Noor. And since then, she has been a faculty member at Western University, which is a large university in Ontario with a correspondingly large biology department. And so she has brought her quantitative genetics background into the realm of speciation throughout her career. And she's reported both very novel empirical results and also advanced new conceptual models in the field. And her lab's work has involved lots of creative fly experiments. But now, as you'll see in this talk, she's even recently layered on top of that a whole new neurobiology toolkit as well. And so I'm very excited to hear more about what Professor Moerhing and her lab have been up to in recent times. And I will turn it over to Amanda now.
Thank you for that really lovely introduction, John. I appreciate that. I'm just going to share my screen. Just a moment. Oops. All right. So hopefully everyone can see that. Does it give me the option to? Okay, I don't see how to make that happen. I was going to change, sorry, I was trying to change my my view, but it's not. That's all right. All right. So thank you so much for inviting me to come and give a seminar, and to the rest of you for coming and listening to this seminar. So as John mentioned in the introduction, my background is heavily on the genetics of things, multiple things. Most strongly within speciation, what genes underlie isolation between species, both in terms of behaviors, the genes behind behaviors, and the genes underlying sterility that arises after species mate. And more recently, I've taken some of the work that my lab has done over many years, looking at the genes, and tried to get the next piece of that story, which is going from the genes to the neurons that those genes are affecting. So the main trait that my lab works on is female rejection behavior. And this trait is really important for in a variety of contexts outside of speciation. So within a species, even female preference behavior is a critical component of fitness and reproductive output of a species. And so often, in the majority of instances, it is the female that decides whether or not copulation occurs when you have a potential pairing between a male and a female. And yet, interestingly, historically, the vast majority of studies that have looked at the genetic and neural basis of mating behaviors have heavily focused on the male component of this interaction. And the piece that I'm most interested in is the female side of that. So what does this look like? So I'm just going to give a silly example here of that we have this Victorian woman and this Victorian gentleman, and he is courting her, and she's receptive to that. So she's sitting there, she's hearing what he has to say, and giving him perhaps some cues that maybe she's not going to turn her back on him. She's going to be receptive to what his advances are. That same female, when paired with a different male, could have a very different response. So in this case, the male is orange, color not chosen at random, and she finds this male repulsive. Now, this male might be fantastic to a different female, and a different male might be fantastic to this female. So whether a male and female are a good male or a good female depends on the context of those two individuals. So no individual is inherently bad or inherently rejecting or inherently receptive. It really depends on the context of how those two come together. Now, of course, these preference behaviors aren't just within a species determining whether copulation occurs. They're also critical for maintaining species boundaries. So this armadillo is a fantastic armadillo that all the female armadillos are receptive to his advances, but no matter how hard he tries courting this Victorian woman, she's having none of it. And so these cues of whether an individual is an appropriate mate or not an appropriate mate are sort of layered on top of, or in addition to, a subtext within that of how good of a mate is this. So are they appropriate? Are they even in the realm of consideration? And then if they are in the realm of consideration, how good of quality? And we've often thought of those as kind of these separate sort of questions, but in reality, it's a continuum, right? That an individual can be so not attractive that they're on the far end of that continuum because they're actually the wrong species. And so my lab group is very interested in both of these questions, both within and between species. What is the basis of that female preference behavior, causing her to be either receptive or rejecting towards a particular male? And for a very long time, this was focused on the genetics of this trait, particularly between species. So what causes a female to reject a male who is the wrong species? And this is inherently painful because doing genetics between species, the traditional way of doing genetics for quantitative genetics is that you make recombinant offspring, and often by definition, you don't have a good time trying to make recombinant offspring between different species. They don't want to mate, or they don't produce offspring that are fertile. And so this has been a really challenging thing to get at, this question. And we're finally at the point where we're starting to get genetic loci for this female preference behaviors. And what I'd like to do now is take that entire arc from gene to behavior. So what are the polymorphisms within a locus that give rise to variation in female preference behavior? What brain regions does that gene influence? What are the individual neurons and neurotransmitters that are involved in that process? And then how do we get variation at each of those steps that then leads to a variation in behavior output? Why is one female receptive and a different female paired with that exact same male is receptive? So we have these very different responses to the same male. And why is that? I'm not working with armadillos, although that would be fantastic. I'm working in flies. So I use flies for all the reasons that anybody would use flies. It's these fantastic genetic tools, of which I'll talk about some of them today. For the question of species isolation, what makes them particularly tractable for that question is that there are crosses that we can perform in the lab between closely related species that we can get to go in the lab, that we can actually form offspring. And while this doesn't always lead to fertile offspring, because of these tools, we can often do things within that F1 generation that allow us to get at these genes. And then they have these fantastic stereotypical courtship and mating behaviors, which allow us to get great repeated measures of what are the key differences in the traits that we're interested in. And so typically when people have looked at Drosophila mating behavior, this is like a classical canonical image of when people talk about Drosophila mating behavior. What does this look like? And you see here that the male orients to the female, taps her, there's an exchange of chemical cues there, extends a wing and vibrates it in a courtship song. He'll then lick the female. This is another transfer of chemical cues, and then attempt copulation. If he's not successful, he'll return to an earlier stage. And it didn't dawn on me until quite a ways into looking at these questions of mating behavior, how this entire diagram of Drosophila mating behavior says nothing about what the female is doing. This is actually Drosophila male mating behavior. This is not a diagram of what actually is happening with both sexes in this mating behavior interplay. And females have a lot of behaviors that they perform that are just as obvious as what the male is doing. So when the male approaches the female, she'll often flick her wings as a sign of rejection. She curls her abdomen away from the male if she's not interested in his advances. One of the most visible things that she can do is called decamping, and that's simply she walks away. So instead of staying stationary, which is what she would do if there was not a male advancing, when he approaches her and she's not interested, you'll see that she'll run away, and the male will follow her then to continue his courtship. She will often kick at the male, either to the side or behind herself. She'll sometimes groom her hind area, and that makes it very hard for the male to attempt copulation. When she is receptive to the male, she will decrease her locomotion, so she'll allow him to approach. So the male can't copulate with her unless she's become stationary. So if she's moving, he can't engage in copulation. And then lastly, if the female has already mated previously, so only mated females can extrude their ovipositor, but if she's in this fashion during mating, But if she's mated previously, she'll extrude her ovipositor, which again prevents copulation by the male. And so for the male to copulate, she has to stop her locomotion, she has to spread her wings, and she has to spread her vaginal plates. There is no forced copulation in Drosophila. And so those female receptive behaviors have to occur for copulation to occur in Drosophila. So I've shown you this cartoon diagram, and here's what it looks like in real time. So there's 2 males and 2 females in this diagram. So here's the male behind, he's courting the female. You see he's got his wing that he'll extend vibrating, and you can see the female keeps changing position around the male. So here she's doing a little grooming. You saw a kick there as he came over to the side. This is another male and another female who just happen to be in the same vicinity. So she's staying stationary right now as the male is doing his song, but now she's pushing at him with her forelegs, and then she pauses for a minute, and then he chases her, and eventually they'll have successful copulation, which occurs here. So she stops moving, he curls his abdomen, she has spread her wings, and then copulation successfully occurs. So that's what it looks like in real time. Here's what it looks like in our actual assay chambers, that we have these multi-well plates that we videotape. So up here, sort of on the top is a copulating pair, and the rest of these, there's courtship ongoing where the male is following the female. The female almost never approaches the male. It's pretty much always the male approaching the female, and then he'll chase her around. Copulation is almost impossible to miss because they copulate for an average of about 20 minutes, which is not bad for a little fly. And so it's great that we can get this very concrete measure of not only the behaviors in between, but unlike some species that have extremely rapid copulation, the copulation is incredibly easy to score as well. So whether or not mating successfully occurred is really easy to quantify. And so we've been interested in the genetic basis of this behavior, and we've got these genes that we're interested in pursuing how they affect neurons. And because female receptive behavior has been almost entirely ignored at the neural level, we don't have candidate regions, really, of where in the brain we should be looking for where these genes would be having their effect. So we decided as a lab that we were going to try and do what I call neural mapping, that we would try hyperactivating and silencing subsets of neurons in the brain to see if we could narrow down what regions of the brain are interesting in terms of female receptive behavior. So here we have a fly head, and in blue behind it is a cartoon of what the brain looks like in the fly. So we're just going to look at that brain, and it's made up of different zones within the brain. So I've just used cartoon diagrams here to show what they are, but they have actually physical structures to them that have that are a certain shape that we can actually see in a microscope. And these have been given names for different parts. So we have, different aspects of things called the mushroom body, the fan-shaped body, the subesophageal ganglion, et cetera, et cetera. So we have all these different zones of the brain. And what we did is use a system to activate or silence different zones within the brain and look at their effect on behavior. And so here I've just sort of pictorially represented roughly the areas of the brain that we were interested in. These 2 large regions on the end here are the optic lobes, and we did not look at those. And we hyperactivated or silenced these. And we did these using this fantastic system within Drosophila, which a lot of you who are in genetics are going to be familiar with this, but if you're not, I'll give you a brief primer on what this system is. So it's a 2-part system that you have a GAL4 protein that is expressed based on a nearby promoter or enhancer. And that protein will bind to an upstream activation sequence called the UAS, which will then, when bound, activate the transcription of whatever transgene is adjacent to that UAS. And because it's a 2-part system, you can have the pieces of that GAL4 under the expression of different promoters or enhancers. You're expressing GAL4 in different tissues based on which strain of flies you're looking at. And then you can have different genes that you're interested in paired with the UAS, and you can combine these in any combination to drive whatever gene you have of interest in whatever tissue of interest that you're curious about. And because it's a commonly used resource in fruit flies, we don't have to make these, we just order them up. So you can say, hey, I want to test these 20 different brain regions, and you order up a GAL4 for each of those 20 regions that expresses in those, and you can just pair them with your gene of interest. In our case, the transgene of interest is either this gene, which is called TRPA1, which is an ion channel. So when you have expression of that, it causes neurons to be hyperactivated at high temperature. So it causes an influx of ions, which causes the neurons to fire. And so you end up with activation, but it's temperature gated. So you have flies not having it activated normally until you put them at high temperature. We then had a separate one for silencing. So this is called Shibire. And this also the one we used as a temperature sensitive version that affects the recycling of neurotransmitters. And so when you have this, again, at high temperature, it causes the silencing of those neurons. What's really great is that this system has more recently been broken now, instead of in 2 pieces, into 3 pieces, where that GAL4, instead of just being made by a single promoter, its binding domain and activation domains have now been separated and put under different promoters. And so what this means is that you can actually have the expression of, say, the DNA binding domain separately using a different promoter express the activation domain. So these have different regions that they're expressed in. And it's only that region of overlap that you get expression of your transgene of interest. And what this allows for is a much finer control over where your gene of interest is expressed. So most promoters or enhancers aren't expressing in a single neuron. They're going to be expressing across a wide range of neurons. But using this split GAL4 system, we can actually target expression down to the level of single neurons in many cases. So it gives us an incredible level of control over where we're having expression. But to start with, we didn't start with single neurons. We started with these larger brain regions and hyperactivated or silenced. And one of the most interesting parts of this to begin with for me was how much of the brain you can hyperactivate or silence, and the flies still seem pretty darn normal. So I was worried that if we'd hyperactivate or silence some really large regions of the brain, we'd just end up with a bunch of twitching flies that are fallen over on their sides. And shockingly, that actually is not what happens. So they're pretty robust to having some pretty large portions of the brain hyperactivated or silenced. And what we found in doing this is that only a particular region of the brain called the mushroom bodies had an effect on receptivity, and that was only when we silenced it that we saw that effect. Now, this isn't to say that other brain regions don't have an effect. Indeed, I'm pretty sure they do, but doing it in this blunt force, large scale way did not reveal those effects. And this is likely because what we've learned, and I'll show this a little bit later, is that you can have neurons of opposing effects. And if you're doing an activation or silencing at the same time, you end up erasing your ability to detect that effect. And so you need to, if you want to get at neurons within those regions, you need to do it on a much finer scale. But starting with this large scale, we ended up with these mushroom bodies. And these were actually, of all the brain regions, they were the only region that we thought maybe is a candidate, because these are involved in higher order processing. So these are known to be involved in male courtship behavior. They're involved in things like learning and memory. So we thought these would be a good candidate for female receptive behavior. Indeed, that is what came up in our assay. Now, these regions of the brain can be further subdivided into a smaller subset of neurons. So in this case, the alpha beta, alpha prime, beta prime, and gamma lobes. And when we can hyperactivate or silence each of those individual, we actually found only the alpha beta lobes had an effect on the female receptivity. And that was, again, only when we silenced them. Now, over here on the left, we have the brain stained. This is an actual brain where all the neurons are stained with this magenta color. And then those mushroom bodies and their processes are stained with GFP. So they're green. Since we figured the mushroom bodies are having this impact on receptivity, we also were able to test all of the modulatory neurons that influence those mushroom bodies. So here we wanted to see if we could figure out what was giving the signal into the mushroom bodies. And then we're hoping to then get to what are the signals for the neurons coming out of those. So we started with all of the subsets of neurons that are known to be modulatory for which we were able to order. So we're missing some because they didn't have a GAL4 driver for them, but here are all the modulatory neurons that we had the ability to test. And so we tested all of them, again, hyperactivating them or silencing them. And we found a strong effect of 2 of these subsets. One of these was the octopamine neuron, OABPM4, and the other one was another neuron called SIF amide. And I'm going to focus on the octopamine because it's a very strong effect. So the effect from the SIF amide neurons was a less strong effect. So still strong, statistically significant, biologically significant, but we had a very strong effect from this octopamine neuron. And it's also a single pair of neurons. So we're hoping this will assist us in doing circuit mapping to get from the origination of receiving whatever signal it is all the way to the output of behavior. So here's what that pair of neurons look like. So the cell bodies are down there at the very bottom, sort of by where the chin is. And then you have all the branching of the neurons which go up, and you can see that process up here. This is where it interacts with those mushroom body lobes. And so what does this actually look like when we look at the behavior? So here we have our, in pale blue is when we have silenced those neurons in females. The dark blue are the two different controls, and these are just the controls of flies that have only the GAL4s or only the UAS and the transgene. So these are controlling for that genetic background. And what we see is that within our assay time, we have a significant reduction in how many females mate compared to the controls when we silence those neurons. We've done observations of these, so we've scored for female rejection behaviors, and the females are actively rejecting the males throughout the assay. So they're kicking them, they're decamping, they're flicking their wings. So this isn't just that females are not mating because they're completely immobile and not doing receptive behaviors, they're actually showing active rejection behaviors. And we can look at this over time also. So again, I mentioned that this is temperature gated, so this tool. And so what we find is that here we have a cumulative score of copulation. So the controls very early on, by about 12 minutes in, the half of them have mated, and by 30 minutes, we have about 60% have mated within this particular assay. And you can see our controls at the bottom in that pale blue. We only have about 10% had mated by about 30 minutes. Because it's temperature gated, we can take those exact same females and switch them back to room temperature. And so this now turns off that silencing. And so you can actually have a recycling of those neurotransmitters again. And when we do this, we see a rescue of behavior. So after that time period, we instantly get females being receptive. It's interesting to me how quickly that effect happens. So females are often primed by courtship. So males court for a certain length of time before a female becomes receptive. And it appears that that time while females were rejecting, that it's still sort of accumulated into that priming time, because you can see very quickly we get a high percentage of those assay females being receptive. It's not a gradual curve like it is in the dark blue at the beginning. So it looks like that priming period is occurring even though females are actively rejecting. Now, I mentioned earlier how it's important to have individual neurons that you're testing versus a large number of neurons, because you can miss effects when you're doing large numbers of neurons. And we see that in this case as well. So this is a single pair of octopamine neuron that when we silence them, we see this very strong effect on female receptivity. We can use a separate driver. So I'm showing it up here on the right. Apologies that it's a different color scheme. This is sort of the laziness of not wanting to make a whole new figure just for a talk. But it's that we used a driver instead of driving in just those subset of neurons. We used a driver that drives in all octopamine neurons, including the OAVPM4s. And when we do that, we do not see a significant reduction in female receptivity. So you can see that the two controls, which are in gray, have statistically the exact same mating rate as those that have all octopamine neurons silenced. And so again, this is just a cautionary tale that when doing sort of groupings of neurons, it doesn't actually let you rule them out if you don't see an effect, because you might be having some sort of conflicting messaging occurring within those neurons when you have more than one being silenced or activated at a time. Okay, so we did this trying to get down to candidate regions that we can then look at for these genes. And so now that we have some candidate regions, where are those rejection genes in females expressed? And then how can we try and relate these back to each other so we can get these to meet in the middle about the genes that we're interested in, the polymorphisms, and then into the neurons that we're interested in. And I'm not going to go into the background on how we did this, but you're welcome to ask me questions about it if you have any questions. But we had previously done a large series of genetic maps trying to find genes, individual candidate genes for female receptivity. And in this genetic mapping, we came across the fruitless gene as a strong candidate for female receptivity. And so this gene is famous in Drosophila land because it's known to be involved in the sex determination pathway, and sort of more notoriously, it's known to play a key role in male courtship behavior. And so this gene, when it has sex-specific transcripts, and when you express female-specific transcripts in males, or if you simply ablate the male-specific transcripts, you end up with males that are incapable of discerning what appropriate courtship targets are. And so they'll court other males and they form these long courtship chains, which are quite interesting to watch. And that's part of where the name came from, that their courtship in that circumstance is fruitless. And so this gene, much to my dismay, is a monster of a gene. So it's about 180 kb. It has at least 5 known transcriptional start sites. It has about 15 or more, so 15 confirmed splice variants. So it's incredibly alternatively spliced. It is riddled with enhancers. We don't even know where the promoters are. It is super painful. And my poor PhD student who started on this had, towards the end, he's like, if I'd known, I probably would have picked a different project. So it is a really painful gene to work on. But through some very careful targeted studies on this gene, we ended up identifying the set of transcripts that start with the P2 first exon. That subset of transcripts is what affects the female receptive behavior. And this was really exciting because this gene has almost entirely been studied for its P1 subset of transcripts. And this is because that's the subset that's sex-specifically spliced. It's what's known to be involved in the sex determination pathway and affect male behavior. And the rest of the transcripts have been almost entirely ignored in terms of what their effects are. So to have a gene that is really complex and affects both male behavioral courtship traits and female receptivity traits has all sorts of implications for questions around the evolutionary basis of these complicated interplay between male traits and female preference and their coevolution. So we figured out that it was a subset of transcripts, but it's extremely complicated to figure out where are they expressed and then to control things within that subset of cells. And the reason why that's incredibly complicated is that all but that first exon are shared with the other transcripts. And that first exon is really tiny. So there's actually only 8 amino acids that come from that first exon. And we looked into it and were told very convincingly by multiple companies that we would not be able to get an antibody that was able to sufficiently target based on that small of a unique amino acid sequence. People, not us, but other labs had tried doing targeted in situ hybridizations and found that they just couldn't get a good signal from P2. And I'm not going to show this data, but we've discovered that it's perhaps because there is a bit more variation in that P2 first exon than was originally thought. So anyway, so those traditional ways of trying to get out where is this subset of transcripts expressed were not working here. We also couldn't simply do the classical thing that people had done in the past where you put a GAL4 next to your transcript of interest, and then you just express that GAL4 using whatever promoters or enhancers are nearby. And the reason why that wouldn't work here is because this thing is riddled with promoters and enhancers, and we don't know exactly where they are. We don't know where the promoter that's causing P2 transcription is located. And so if we put a GAL4 in there, we wouldn't know if it's being expressed in the same pattern as P2. In fact, it very likely would be influenced by multiple things and have a very broad pattern of expression that we would not be capturing properly. And so fortunately, there was a newer technique that came out very recently that's called the Trojan system. And I'm not going to go into, again, the mechanism of this in detail, but if anyone is curious, you can ask me. I can give you some gritty molecular underpinnings of this. But essentially what it does is it lets you stick something in that tricks the machinery into thinking it's an exon of your transcript of interest, allowing it to be incorporated into that transcript. And then that GAL4 protein is cleaved off at the protein state. And so you end up with only making GAL4 protein in just those tissues where your specific transcript was made. So I had a fantastic grad student who had really good molecular skills, who was able to successfully create this complicated system within this fruitless gene for that P2 set of transcripts. And so here's what we found when we paired that P2 driver with, again, that UAS transgene that allowed for hyperactivation of neurons. So I'm just going to show this short video. So in the middle are the ones that are test females. And we have a female and a male that are separated by a divider at the start of the assay. And on the end are our 2 controls. And this is just a sample of what happens. And this is shown in high speed, so the flies do not normally move this fast, simply because you're going to see as this goes on that the controls on the 2 ends, even in just this assay, that they both mate. And those two in the middle, which are our hyperactivated females, the males keep courting and courting, but the female keeps rejecting, and we never see copulation. So here at the end, they've copulated, and in the middle, they'll just go on and not copulate. And indeed, we see an extremely strong effect of hyperactivating these neurons in females, that we see almost no mating in those that have that subset of neurons where you have hyperactivated them. And again, these neurons are neurons that express that P2 transcript in female brains compared to the two controls. When we silence those neurons, we didn't see an effect. And again, this is with that inducible system where it's temperature gated, and we did not see an effect on female receptivity. But we decided that we would follow this up with a constitutive silencing of neurons. And so the difference between this is with the inducible system, the neurons are firing normally throughout the lifespan of the female up until we take an adult female, put her at temperature, and in that moment, those neurons are silenced. But it's possible that silencing neurons early might impact their connectivity in such a way that having those neurons not be functioning properly influences later behavior. And so that it's not a situationally silenced that affects behavior, but rather if it's a developmentally silenced subset of neurons. And so we used two different constitutive drivers that are expressed throughout, where these neurons are unable to recycle their neurotransmitters throughout the life of the fly. And in both of these, we saw a significant effect on female receptivity. So this impacts the connectivity or the development of adjacent neurons that then leads to this effect of not having females be receptive when they're adults. So what we find is that there is a strong effect of these neurons that express P2 on female receptivity, and we can now control and target how these neurons are firing in order to see their effect on behavior. So our current assays, of which I have zero data to show you because they're going on exactly now, is instead of expressing a hyperactivation or a silencing, we're using that same system, but paired with a UAS RNAi, so RNA interference, for different neurotransmitters. So we're going to remove the neurotransmitters for different subsets of neurotransmitters within those neurons to figure out what kind of neurons these are. Are they an ectopamine neuron? Are they a dopamine neuron? What kind of neurons are these? Again, to give us insight into what the downstream targets of them would be. All right, so where are these in the brain? So this is, as John mentioned, this is new for my lab, this area of moving into neuroscience. So our brain imaging is not that fantastic. Those previous images I showed you were done by other labs. So this is a low impact, so this isn't a confocal image. This is just done on through our regular microscope. But nonetheless, we've used that with a decent accuracy to be able to figure out where these P2 neurons are. So here we have a brain. This is not counterstained with pink, so you can just see the faint outline from green autofluorescence of this brain. And we can see which subset of cells are actually expressing that P2 transcript. We're in the process of getting better images now. And if we draw a schematic of the brain, we see that the lamina, which is the outer layer of the eye where you have the optic nerves, that fluoresces. The ocelli, which are another optical cell type, and then the cantle, and then these little small cluster of cells down in the subesophageal ganglion. What's really interesting is if I go back to that OAVPM4 image, again, that was done by another group that made the lines that we used, we see that same region of the brain appears to be harboring cells that are involved in female receptivity. Now, are these the same cells? Well, we haven't done that test. I would say by our results that they're not likely to be the same cells. And I say that because when we hyperactivated OAVPM4 neurons, we did not see an effect on female receptivity. Instead, we saw that effect when we transiently silenced them. Conversely, with the FRuP2 neurons, when we transiently silenced them, we didn't see an effect on behavior, and we only saw that effect when we hyperactivated. So it appears to be that they're not the same neurons, but they are located in the same region. I can't say definitively also if that particular subset of neurons is what's causing behavior in those FRuP2 females. So we have to go down and map this down further, which we're in the process of converting our Trojan P2 into a split GAL4 with the P2 so that we can combine it with other drivers in order to reduce the number of cells where that's expressed. And all of that is ongoing. So I'm not to the end yet, so I don't want you all to think like, oh, this is the end. But so far, what we found is that there's a locus that we found that seems to affect female receptivity, and we figured out what transcript is affecting. I didn't go into the details of this, but we've actually are in the process of testing which polymorphisms in that FRU-P2 exon are underlying that variation in female receptivity. But that's, again, ongoing. We found that there's certain regions of the brains and indeed certain subsets of neurons that appear to have a strong impact on female receptivity. And for the last part of my talk, what I want to talk about is if there's an overlap between courtship and rejection pathways in males and females. And this is based on some very unusual results that came up while we were doing our neural mapping experiments. So in looking at different brain regions, I had this great undergrad, Andrea, in the lab who said, okay, for my honors thesis project, instead of doing brain regions, I want to do cells that express certain sex determination pathway genes, because that might be another avenue that might get us some candidates. Since fruitless is involved in the sex determination pathway, and we know that affects behavior, maybe other sex determination pathway neurons, they're affected by these sex determination pathway genes. Maybe those also have an impact on female receptivity. And she wanted to focus on these neurons that express a gene called double sex, which is also involved in the sex determination pathway. And so she tested different drivers that hit on different subsets of neurons within that cluster. So in orange are all of the double sex neurons. You can see there's not a ton of them. And she tested multiple different lines, again, that have that overlap of expressions. You can get a subset of neurons. And in those, there was one that expressed in this particular subset. And when she hyperactivated them, she found that she got no mating. But fortunately, she was a very observant student. So she didn't just look like, was there mating or not? She was watching what was happening. And she comes to me and she said, okay, they're not mating. I'm like, oh, cool. And she said, yeah, it's because the females are kicking the crap out of the males the entire assay. And so indeed, when you hyperactivate this subset of neurons, it's not so much that females aren't receptive, it's that they become hyper aggressive, and the males simply can't court and mate the females because the females are aggressive towards the males during the entire assay. So this is what happens when you're thinking you're going to work on female receptivity, and then you have a, that's funny moment, and you end up making a whole different project because it sucks you down a whole different avenue of research. What's really, really cool about finding this by chance is that we know almost nothing about the neural basis of female aggression or genetic basis of female aggression. Almost all aggression studies that have looked using genetic and neural model systems have looked at male aggression. And this makes sense in some ways because it's easier to get males to be aggressive within the laboratory than it is to get females to be aggressive. But it also means that we know almost nothing about whether this is sex specific, whether females which are slightly less aggressive than males, is that just because they have a bit less than what males have, or is it a different pathway? And we can't answer that if we don't have any idea what underlies female aggression. What's great is that these neurons do not cause aggression in males. So if we hyperactivate these neurons in males, they don't cause the males to be aggressive. So they're specific for female aggression. So Drosophila aggression has almost entirely been studied in males. Very, very little has been done in female. I think I found 3 papers that mentioned female aggression in some way, and 1 that actually looked at it directly. So there's very little on female aggression. But female aggressive behaviors and male aggressive behaviors are largely similar, just to different degrees. So we see lunging, where they'll lunge at each other. Sometimes they'll fence or box, where they tussle. There's a lot of wing threats. They'll do a lot of wing movement when they do that. And females have behavior also that's called headbutting, where they'll essentially ram the other individual. And what we've found so far is that what behaviors are shown largely depends on the context of what individual they're paired with. And we think this is also true for males. So the reason why we see some differences in male and female aggression is largely based on the response of their partner. And so a male, if paired with a female, is going to court her. A male, if paired with another male, they'll escalate because the other male will respond with aggressive behavior. When a female is aggressive towards a male, he'll try to court her, he'll respond. If she's aggressive towards another female, that female doesn't tend to escalate. She'll tend to just walk away. So we see different escalation of aggression because of how the partner responds. We also find in females a lot of chasing, where one female will run away, or the male will run away from the aggressive female, and the female will simply pursue. And so we see a lot of chasing and pursuing behavior. So a reminder that this is what regular male and female pairings look like. This is that same courtship video I showed earlier where you have the male approaches the female. So males here are a little bit smaller than the female and they have this black butt. So that's how you can tell them apart. So you can see females are kind of just walking around, but mostly if there's one following another, it's the male. So the male's following the female here. And now we're going to be looking at females that have that subset of double sex neurons hyperactivated. So we can just focus here on this bottom right panel. And here, this again, this is a female. You can see she's got sort of this paler abdomen. Here's another female with a pale abdomen. And we'll see what happens when we pair those hyperactivated females. And you can already see the females approaching the male. She's pushing at him with her forelegs. She's headbutting him. You can see some wing flicking that she's doing. Again, over here, we see this female's approaching that male. Here, the female's approaching the male, flicking her wings at him. So we see very strong difference in behavior between what we saw when we have females that don't have these neurons. We can see this most pronounced in what my student called the cage fight, where you pair two females that are both hyperactivated. And this is what I mean by that partner having an effect on what happens in terms of aggression. So these females are both being triggered to be aggressive, and you can see that tussling, that headbutting, they're pushing with their legs, and they will do this for hours if we leave them this way. So they will fight very strongly for a very long time when we hyperactivate these neurons. I also want to point out that this is very clearly not females behaving as if they're doing courtship. So these females are clearly showing aggressive behaviors with the headbutting, the pushing, the tussling. And this is something that's awkward that I have to even justify that this is actually aggressive behavior. There's a strange pervasive culture within Drosophila that if females do something that even remotely looks like what male courtship looks like, such as approaching or wing flicking, you have to justify how that's not courtship because it's presumed that it's more likely for a female to show male courtship behaviors than for a female to show female behaviors like aggression. So I find this a bit strange, but there it is. So hopefully you can see this is very clearly not courtship, this is aggression. And we can see this when we quantify compared to our controls. So we have that transgene, the GAL4, each alone, and then when we pair them together. And this is just a tally of all of the different aggressive behaviors. So we did score these separately as well. But if we just sum those all up, we see almost no aggression at all in our control flies. And a huge number of aggressive encounters within those that have these neurons activated. So this assay was a 10-minute assay, and we see between 70 and 90 aggressive encounters depending on who she's paired with. So a huge amount of aggressive interactions. Again, and who she is paired with in this case is simply a male, so a wild type male. So these are the level of what we see, or a wild type female. So these are individuals that are not hyperactivated, they're not responding in kind. You do notice, however, that these are slightly different. So there is more aggression when she's paired with a male than when she's paired with a female. So note that these y-axes are different. And we wanted to start getting at what the basis in terms of context was for these female aggressive behaviors, because nobody's looked at this before. I mean, there's very little done on female aggression. And so if I'm just using, because I think it's sort of easier conceptually to follow, check marks to display the amount of aggression that we're seeing. As you saw in the previous graph, we see a high level of aggression with females, but an even higher level when paired with a male. When we take two females that are both aggressive and put them together, we see the highest amount of aggression. So we thought, okay, this seems to be influenced maybe not directly by sex, but it seems to have some influence on there. That's influencing it, that there's some influence based on who she's paired with. We wondered if it was maybe species specific. So if we pair with a male of a closely related species, so in this case, Drosophila simulans, will we still see aggression? And the answer is yes, but not quite as high as with a conspecific male. Now, one thing that we thought about when looking at this data is in these pairings, the individual that they're paired with has different amounts of locomotion. So a female paired with a female, that female that's being attacked doesn't move nearly as much as a male because that male is continually trying to circle around to court the female. With the D-simulans males, he doesn't turn around and try and court as often because he's the wrong species. So he will court, but he's just not quite as interested in it. And then the aggressive female has the most aggression because it's like game on, head on. So we thought maybe that locomotion had an impact on this. So we wanted to look at that movement and see what happens. And so we paired females with an immodal male. This is a male that's decapitated, or with a hyperlocomotive female. And so the hyperlocomotive females are females that have a transgene that leads her to move much more rapidly. And so here, this is my favorite video of all. Here's females, capitated males.
Yeah. Oh, thank you. Okay.
Anyway.
Thank you.
Okay. So she's paired with these decapitated males, and I'm just going to rewind it a little bit because I was distracted. And you can see that she is beating up those decapitated males. So here's the female approaching that decapitated male, and she is pushing him and grabbing him with her forelegs and generally showing really strong aggressive behaviors towards him. I'm not showing the video of it, but the hyperlocomotive female induced equal levels of aggression as what we saw with wild type males. So if a female moves more like a male does, she gets more aggression. So we see a very strong relationship between the amount of locomotion and how much aggression there is, but it's not required. So if you have an immodal male, we still see aggression. So the movement enhances aggression, but it's not required to induce aggression. So we thought maybe it's very strongly visual more than anything then. Like maybe that's all you need, like that immodal male, maybe it's just a visual cue that triggers her to have some aggression, and then the locomotion enhances beyond that. So if you place her in just an empty chamber versus if you place her with a white object, in this case, it's like a teeny piece of white filter paper that we rolled into a fly-sized burrito, or a black object, which is the same, but with black filter paper. And none of those induced aggression. So if we put aggression in there, or an aggressive female in there, she did not care about these objects. So the visual cue alone is not sufficient. Based on our reading, we thought perhaps there needed to also be an olfactory cue. So a female is not going to be aggressive towards any old object. She needs to think that maybe it's a fly, that maybe it's something that's a competitor. And there's a chemical compound called methyl palmitate, which I'm abbreviating MP here, which is an aggregation pheromone, and it's also found on Drosophila simulans. So we thought maybe this is the chemical cue that induces females to think of an object as a potential target for her aggression. And indeed, that's what we saw. So we would see aggression when we add the methyl palmitate. And so here's what that data looks like. So here is that, again, that UAS by itself, the GAL4 by itself, and when we add methyl palmitate, we see no or extremely tiny amounts of aggression. If we take our activated female and add her with the visual cue, in this case, it's the black little burrito for each of these, we also see no or almost no aggression. And then when we add that methyl palmitate to it, we see an induction of aggression. You can see it's not quite as high as what we would see with an actual fly. So we don't see, 70 aggressive encounters. We see a smaller number, but we do see a significant amount of aggression when we have those combined. And so it seems to be that females need a pigmented visual cue with methyl palmitate as a chemical cue to trigger her to consider an object as something worthy of her aggressive encounters. We are now in the process of testing the neurotransmitters, just as I mentioned earlier, for behavior. We've done a first pass assay of some neurotransmitters, but we're in the process of repeating them. It seems, I'm not going to show the actual results of this because it's still extremely uncertain. I need more data, but it looks like maybe dopamine is involved, but we need to see that more clearly. However, we have some really lovely results on looking at what receptors are potentially being affected. So for your neuron, you have both the neurotransmitter it's releasing, and that's affecting that downstream signal of what neuron it's talking to, but you also have whatever cue it's receiving. So it's got receptors for other neurotransmitters that are what are causing it to be activated. And so what we did is we knocked down those receptors one by one to see if any of them influence the female's ability to respond in terms of aggression. And we saw a strong effect of GABA on this. So GABA is a suppressor. So it's typically a neurotransmitter that causes a silencing of neurons until it is removed. And indeed, that's how we see it here. So here is I'm just showing one of the controls here where we have no aggression. Here is our hyperactivated females, which have a high amount of aggression. And if we take away GABA, we see an extraordinarily high amount of aggression. So in this case, it was about 250 aggression encounters within a 10-minute assay. When we break this down into individual aggressive behaviors that a female shows, we see that it has an influence on all of them. Statistically, only 2 of them were statistically significant, but you can see that every single component of that aggressive encounter was increased compared to the controls. So we see a huge increase in female aggression when we remove GABA. So this suggests that these neurons typically are being suppressed by GABA, And then when GABA is removed is when females will display an aggressive response. I'm going to end now on a sort of a cautionary tale on why it's so important to have context when you're thinking about working on a trait in one sex that has historically always been worked on in another sex. And this is because it heavily flavors how data is interpreted, particularly when working on behaviors. So these same neurons, these exact same neurons, it's actually using the exact same Gelfer driver that we've been using for these studies, have been studied in males, and they found that it affects male courtship. So when I mentioned when we hyperactivate these neurons in males, it doesn't cause aggression. The really interesting side note to that is it causes the males to court instead of be aggressive. And the people who worked on this is about 5 years ago now, they then took these neurons, they said, aha, they affect male courtship. And they tested them in females to confirm that those are male courtship neurons. And they saw females approaching, moving their wings, and said, these are male courtship neurons because look, we induce male-like courtship in females. And again, it's this caveat of that when you're activating neurons in a female, your default assumption should be that maybe they're doing female things, not that these must be then making them into the opposite sex. And so this was a mistake by these researchers to think that these neurons are male courtship neurons that activated in females make females behave like males. And indeed, we've even looked at the videos that they posted of this and we're like, yeah, that's aggression. They just really were looking at it through a lens of male courtship. And so they interpreted those behaviors as male courtship behavior. So I just want to throw that out there as a caveat, especially for those of you who work on behaviors, to be very cautious in how you interpret your behaviors that you're observing. So just to summarize, female aggression appears to be mediated by a subset of double-sex neurons. It requires some sort of pigmented visual cue plus a chemical cue, methyl palmitate, and increases with the movement of the target, so that who the female is paired with really impacts the degree of aggression that we observe. It's suppressed by the neurotransmitter GABA, and is affected by the same neurons that influence courtship in males. And I have all sorts of questions in my head that I don't have answers to yet of how that would evolve, that difference in males and females of a subset of neurons affecting male courtship in one sex and female aggression in the other sex. That has some interesting evolutionary questions that raises in terms of wondering what came first and how that came to be. So to end, I definitely want to thank the students who did all of this work. So I'll go through and highlight those who did the work that I presented here today. So Priya Mahavir and Brendan Charles did that broad scale neural mapping, and then Brendan Charles and Sebastian Hein did some fine scale mapping for female receptivity. Andrea Bevan is the fantastic undergraduate who did the first identification of those aggression neurons. This was then followed up a little bit by Brendan Charles, but largely by Sebastian Hein. Masa Farmanbar did a lot of the neurotransmitter tests, looking at what are the neurotransmitters that are involved. And then lastly, Will Jung did the work on making that P2 Trojan construct and testing the effect of those P2 fruitless neurons on female aggression. And then, of course, I have to thank all the funding agencies here in Canada, and a little shout out for any of you who are students or postdocs who are thinking about jobs. Canada is a fantastic place to do research. We have consistent funding and parental leave and nationalized healthcare and many other things that are amazing. So please consider coming to Canada if you're on the job market in the future. And with that, I'll take any questions.
Thank you very much, Amanda, for a fascinating talk. I'll start out with one question. Reaching back into the earlier part of your talk about fruitless, I'm wondering, you know, the fact that both female and male courtship behavior, at least courtship relevant behavior, is encoded by fruitless. Do you think that could facilitate coevolution of mating behavior between the sexes due to variants that are carried on the same haplotype, and could that facilitate mating behavior divergence between species or even between populations of the same species?
Yeah, so the answer is yes, or maybe no, right? I mean, there's also a lot of constraints on evolution, especially if you have antagonistic aspects between males and females that can make that really complicated. So I do certainly think it eases, it essentially becomes, although it's not truly the one allele model, because you're literally talking about different subsets of the same gene rather than the exact same exons. But it does lead to essentially a one allele model where you're looking at the ability to have a single locus influencing both sides of that equation, both the male trait and the female trait, which makes it much easier conceptually then in terms of how that can be inherited and passed along. You aren't going to break up these combinations. So it certainly would facilitate, but I don't want to have it be just a simple answer like that, because it also can constrain, right? You can also have difficulty then in affecting regulation of one subset of transcripts compared to another. In this case with fruitless, because the alternative splicing occurs at the 5' end and there's different promoters, I think that bypasses some of that problem, because you can actually have regulatory divergence between that subsets of transcripts, which would be much harder if it was a difference in internal exons. So I think that maybe makes it easier to have that separate selective pressures on the male and female subsets.
Yes, thanks. It definitely makes sense that it will never be quite that simple.
There is, unfortunately. Every time there's something, you're like, oh, more complicated than I thought it would be.
I have sometimes noticed fruitless pop up as an outlier in my population genetic scans for selection. So it'll be fun to talk more about that in the future.
Well, if you ever talk to my grad student, he'll tell you to run as fast as you can from any tests on it. All right, were there any other questions?
I had a quick question.
Yes.
It was a very, very interesting talk and set of data and behaviors, and I was curious for the female aggression, for like headbutting and some of those behaviors. Could you give an idea of how frequently and when females use those in like?
Yeah, so what's the order of events that we see? So we didn't quantify that. So we didn't actually look at, but we have that data because we timestamped the student who scored when the behaviors occurred, actually timestamped them. So we could do that, but we haven't looked at that if, if wing flicking always happens after headbutting or more likely, and so on and so forth. So we haven't looked at that yet. I know there was an older paper, which is one of the three, by the Kravitz lab, that looked at male and female aggression and did look at those components, but their data is going to be different from ours simply because they didn't have any escalation of aggression, because the way they did their assays was they paired wild type females with wild type females, and so they would have these short aggressive encounters that would end right away, whereas ours, since we're artificially triggering them to be aggressive and not turning off that signal, we can actually get the escalation to see if the order of events when you have escalation are the same or not. So I don't know. I'm saying there's some evidence that we could look at from an earlier study, but we haven't done that, and we should, because our information might be different.
Thanks. Very short follow-up. Are there like are there any like non-genetic intervention triggers that consistently generate aggressive behaviors? Like, what did the Kravitz lab induce it with, or was it just waiting?
Yeah, so no. So if you just put them regularly, like you kind of never see aggression. So essentially you have to food deprive the flies ahead of time, so they're a bit starved, and then you give them a very small food resource. And even then with females, you might get 1 out of every 20 times you would get some sort of, so this is why people don't look at female aggression, because it's super painful to get any assays. Males, you do the same, but you also put a decapitated female on the food, so they have an extra resource, because males don't care if the female is decapitated or not, to try and go for, and you'll see aggression much higher. I'm not sure what their rate of it is, but relatively consistently, they can get males to be aggressive when they have that sort of a setup. So it's a bit contrived in that sense too, even then, when you induce it in a laboratory, because you have to have them starved ahead of time, limited food resource, et cetera, et cetera. But they can induce it without having to use genetic triggers, for sure. So we have not done that, because I like the genetic trigger to answer the questions that I want to ask. But we've thought about that for some follow-up things. So when we find certain things using our trigger, we'll go and do the more painful assays then to confirm that they're also similar in a naturally induced state. But we're not to that point yet.
Yeah, that makes sense. And I expect starvation might be triggering it a fair bit more often in the wild too.
True, true. Although I don't know, there's not the limited food source thing might not be as big of a trigger though from melanogaster because it's such a human commensal, right? Like there's not usually a lack of compost heaps.
Thank you.
Are there any other questions? If not, I can be off the, well, I'm not actually off the hook though, because I get to meet with the students next, so.
Yeah, the students may be saving up their
I
questions.
know, they'll save them up.
All right, well, thank you so much for being here and at least virtually and sharing your exciting recent work with us.
So much for inviting me. This way, I'm sorry I couldn't be there in person. It's just it was too complicated. So I appreciate having the virtual option.
Well, we had some nice icy weather yesterday, so that might be for the best.
True, we did too. All right, so do I stay on the same link for the?
Yes, yes, please.
Okay, good.
Students
Okay.
gonna stay here. Thank you so much for your taking time.
And I look forward to staying in touch in the near future.
Yeah, please do. Please do.
All right, take
Bye.
care.
You were saving your question for the student meetup. Look at that, just like right at the front edge.
Yeah. Yes, thank you so much. That was a very interesting, wonderful talk.
Thank you.
Yeah, I'll go. So the one thing that I just thought was interesting, you were talking about looking at the mushroom body and activating that and seeing the changes in the female reception. I'm curious if you know anything or thought about or have any data about effects on the mushroom body like that, because the only real thing I know about the mushroom body is how it's involved in like olfactory sensory input, and like also how important that is for female courtship, like sensing different things from like CHCs or other kind of cues like that. So I'm wondering if you have any thoughts on how that might be affected, like if you're just activating neurons for sensory input that, or superficially underactivating those, if you like think there's some sort of effect that could be having.
So potentially, I mean, the signals are coming in from, there's something that females are receiving that's causing them to do it. So what we found, I haven't done this with the neural hyperactivation of the mushroom bodies, but we did with the OAVPM4 neurons and with the FRU-P2 neurons. We did assays where we removed the last two segments of the antennal lobe. So this takes away the primary input of both olfaction and auditory cues to see if that would rescue the effect of saying, hey, this is a terrible male. And it did not. But I can't say that they don't though completely, because you're doing both a neural trigger and removing a sensory cue. And so there's a compounded effect there that might prevent our ability to detect it. So I don't think it's olfaction in this case that's affecting the female receptivity, simply because we do these assays. So the assays that we were doing with the OAVPM4 neurons, those were within species. So those are with the same males. So the only way I could see that would be impacting is if in silencing those neurons, we're essentially falsely telling the female that it's the wrong chemical cue. But in melanogaster, it would have to be the false chemical cue that is triggering. It can't simply be the absence of chemical cues, because if you have males who are lacking in chemical cues, the females will still mate with them. So you can take these, they're called enocytless males that don't produce cuticular hydrocarbons, and the females will still mate with those males. So it's complicated. So there's no like obvious answer. And I don't know if there's a clever way to get at that, because every way that we've tried to get at it, there's always a caveat or an addendum to the results. So I don't know quite how to get at that aside from in a concrete way, aside from the way that we're doing of trying to simply map backwards to see what input's coming in.
Thanks.
Yeah. Manuel?
Hi, that was an amazing talk. Thank you for that. I was wondering, since fruitless is very important, it seems like in making like behavioral barriers between species. Do you see any like differences between species, like phylogenetically speaking, between flies of different species and this specific gene?
So fruitless does vary between species. Of course, there's sequence variation between them. If you mean for P2 specifically, for that particular subset of transcripts. I have a graduate student right now who's pulling up, which it shouldn't take very long. So it's not done yet, and it should be, but pulling up the sequences to see what the sequence variation is between closely related species. What's challenging with that is, so we're going to do that, but I'm not sure how informative it is until we figure out what it is within P2 that's causing the isolation in the first place. So there's an obvious polymorphism within that P2 exon that causes an amino acid change that actually will change the structural functionality of P2. And we're in the process of using CRISPR to alter that. So to change it from the melanogaster amino acid to the simulins amino acid within the sequence to make those nucleotide changes. But until we know if that actually is effective, it could be regulatory. And the problem with regulatory is that's going to be in the intronic space in between P1 and P2 is the most likely location of that promoter region. And there's huge amounts of variation in there and which part of that matters. And so I think until we know what sequence it is that is causal, then it's a bit of a fishing. Like, we're going to see lots of differences. Are they important? I don't know. Like, I mean, there's going to be a lot of differences that are not important for this trait. So, we can compare the sequences, but I don't know how informative it's going to be without first figuring out what the polymorphisms are that really matter. So it's tricky.
Thank you.
Yeah, Megan, I think was next. I'm not sure actually who's next on my row of people up top.
Yeah, so that was a really good talk. I was wondering, you mentioned like you had paired these aggressive flies with other flies and seen that like they kind of had a preference for color, like when you paired them with dark objects versus light objects.
Yeah, so they didn't show aggression towards light colored objects. They only showed them towards pigmented objects.
I was wondering if you've tried like mating them with different flies, like ebony flies or like yellow flies, and if you've seen a difference in that.
Yeah, so we haven't tried aggression. The only mutant flies that we've tried aggression with were those hyperlocomotive females, which look the same as regular females, but they just move a lot. And then we tried that with simulins males, which look almost identical to melanogaster males, so they're not dramatically different. So we haven't tried. So that's a good thing. We could try it with dark-bodied flies or with yellow mutants, which have a lighter colored body, and see if we still see aggression. I would predict that we would, because there's still enough pigment. So certainly ebony flies have a lot of pigment, so they would for sure. So I anticipate that we would still see a fair bit of aggression. Related to that, though, what I want to do is pair them with flies that are even more genetically distant. So if we pair them with Yakuba, if we pair them with Pseudo obscura, if we pair them with Miranda, will we still see aggression, and how far out can you go before this fly, which is still a fly, is not recognized as an aggression target? And I imagine that it will relate to those aggregation pheromones and when those diverge, because if she's aggressive towards a little black burrito, then I imagine any fly that looks remotely fly-like is going to be suitable as a target. Flies don't have great vision, so it doesn't have to be visually very accurate. So I imagine it's going to be much more chemically based at that point.
Okay, yeah, that sounds really interesting.
Hi, thanks for the interesting talk. I have a question about the olfactory signal that raises the aggression behavior of the female. That reminds me a little bit of, though I might have missed some information about that specific signal, what that does, but that reminds me of the sensory bias hypothesis you mentioned in the hypothetical model of failed preference paper, where there might be like kind of a signal, kind of a stimuli that might have provide some non-mating fitness advantages for the female that might be exploited by the male. And then it's like some sort of cheating, the male cheating to the female, and then the female might evolve some countermeasure to this kind of cheating behavior. So I'm asking about the origin of the evolutionary origin of the counter behavior to.
Well, I don't think in this case the aggression comes from sensory bias, because so in sensory bias you would have that females would have a strong preference for a certain cue that taps into something that she's already sort of primed for, like, oh, maybe I can really see this particular shade of blue, but it doesn't exist yet. And if a male then randomly evolves that shade, I'm going to be like, wow, that's a great shade of blue. With the aggression, what I think the methylpalmitate chemical cue is simply a female being able to recognize that object as a fly. So otherwise, again, they don't have great vision. So they need more than simply a blob of something in their visual field to cue to them that that's actually an object that's not, a leaf or a twig or something else, that it's actually a fly that's worth aggression. So I don't think it's sensory bias in this case. I think it's simply the added information that chemical cue provides. That in this case, that methylpalmitate is a cue that all the flies have already as an aggregation pheromone. So I don't know if I'm answering your question properly, if that's what you were actually asking. But in that case, in the aggression, the methylpalmitate is simply just an aggregation pheromone that already is used by flies for aggregation. Yeah,
I guess that's in the thing underlying the phenomenon is quite straightforward. Well, but the kind of experiment that might be able to answer the question about the sensory bias in female aggression behavior you have to use some olfactory signal mimicking marula, like its favorite fruit, with some black pigmentation vision signal to. That's just
so we could try different odorants to see if there's other odorants, and that's true. We didn't actually try a whole suite of odorants at all. We only tried the methylpalmitate. So it's possible that certainly other cues might elicit an even stronger aggression response, and we haven't tested that at all.
Okay. Cool, thanks.
Is that, I don't know if I'm sure, is it Ceci?
Yes, okay. Thank you for a great talk. So I was wondering if there is any overlapping between the molecular basis of courting rejection and female aggression, because I'm asking this because I feel like when female Drosophila is actively rejecting courtship, it becomes more aggressive. So I was wondering,
So absolutely. So I think these are simply different points on a continuum, because female rejection behavior is literally aggression. Like, she is pushing that male, she's kicking him. It's excuse me. It's a little bit different on that spectrum, simply because she also has a lot more avoidant behavior. So aggression doesn't have avoidant. Responding to aggression has avoidant behavior, but that avoidance isn't part of the aggression suite. Whereas female rejection behavior, one of her rejection is simply getting away from the male so that he can't try to copulate her. So there's a different subset of behaviors within that female rejection that's not in the aggression, and vice versa. But you're right, they're absolutely right. There's a lot of overlap. So a female kicking at a male, flicking her wings, sometimes she'll push it in with her forelegs. Those are the same behaviors we see when she's being aggressive also. So it's female rejecting advances in any sort of way, or another individual in any sort of way. There's an overlap in the suite of behaviors that are involved in that, for sure. So one thing that I'd be really curious, which we're not close to at all because we need way more info to get to this level of question, but is do those pathways intersect? So does the aggression pathway tap into a subset of neurons that's like a Venn diagram with the female rejection behavior neurons? Like, are those actually triggering similar overlapping parts of the brain that have subsets that are different but have a lot of similar shared features? Or are they really separate pathways? And we don't know that because we don't know what the pathways are yet. So we can't answer that, but that's something I'm really interested in. Is I think of these as being on a continuum and part of the same pathway, but they might be completely separate, and we really can't answer that yet.
Thank you. And I really like your videos.
Oh, thank you. Aren't they great? The
Yeah.
females beating up flies. I find them very hilarious, actually.
Yeah.
Siwan, did you have another question?
Oh, sorry, I forgot to lower my hand. Sorry.
Oh, that's okay. Yes, Hui?
Yeah, so I'm sorry that I missed part of your presentation, so I don't know if you have mentioned this in more details, but I would like to hear more about the chosen GAL4 that you used to circumvent some problem with the P1, P2s transcripts.
Oh yeah, so I didn't go into detail, so that's a perfectly fine question. So the Trojan GAL4 system is it contains splice acceptor sites, which essentially trick the machinery into thinking it's an exon. And so you insert it directly next to your exon of interest. In our case, that would be that P2 first exon. And the machinery, when you're having transcription, treats that like an exon, so it incorporates that into your transcript. And then within that construct are sequences that when you've got translation, it causes ribosome stalling, which causes, when it then reaches that, so it's essentially a skipping, that then it has that GAL4 is translated as it and becomes a separate protein piece. And so you end up with a truncated P2 protein from that homolog, because it gets cleaved off and becomes then the GAL4 and this little truncated P2 first bit of protein. And so one thing, just a caveat of that system is if you make it homozygous, you essentially end up not making any protein from your gene of interest. So we maintain it as a heterozygote. But it ends up making that GAL4 protein then in only the places where it made that P2 transcript. So I don't know if that was enough detail for what you were hoping to have, but that's how that system works. So there's a paper, if you like go into PubMed or even Google Scholar and type in Trojan-GAL4, I think it was 2015 is when the first paper came out, and they have diagrams showing exactly how that works and all the different molecular pieces that are involved. It's a complicated system. One of the things that is be very careful of is you have to have it in frame. So you have to make sure that you make your GAL4 in frame with your original transcript, or else you make you won't make the GAL4 protein, you make some other random set of amino acids that aren't GAL4. So there's some nuanced things that have to be done when you make it to make it so it's functional.
So I don't go on Drosophila, but from the sounds of it, it sounds like this approach can be applied to other organisms as well.
It could, I mean, you could use the Trojan concept to have something expressed in, I mean, the reason why this was really useful is because it's making GAL4, and we already have all these fantastic UAS transgene pieces that we can order up, and then we can just combine those into a fly. So if you could, I think, use it certainly in other organisms, as long as their splice acceptor sequences are the same, so that you end up with the same concept of incorporating that exon. And then you have to decide what it is that you want expressed, like what system within your system would make it worthwhile. So in Colloquium, maybe you would just have it where it makes a GFP. And so you can have fluorescence in whatever cells your gene of interest is being expressed in. And maybe that's the thing that you want. So it could be used in other ways than how we've used it here, I think, for sure.
Yeah, that was what I was thinking, actually. Thank you for that. Yeah.
No problem. Yeah. Myron?
Yeah, I don't want to jump ahead of anyone if they have more questions, but I was just curious. You mentioned this just kind of passing in the paper about Frivolous that you sent, but I was curious, the crosses that you were looking at, you were talking about how the hybrids, well, first of all, that the melanogaster or simulans females won't mate with melanogaster males. Is the opposite true as well? Will melanogaster males mate with simulans or no?
Yeah, so melanogaster males will mate with simulans. It's not at the levels of wild type. So they're not like super excited about those simulans females, but they'll certainly, I mean, that's how we make the hybrids.
Right.
Is doing that. So you take, yeah. So you can take, well, wait, sorry, am I doing this correct? You take, wait, which direction is this? Sorry, I'm having a, yeah. So simulans females really don't like melanogaster males, but the melanogaster females are much more permissive. So they'll mate with simulans and melanogaster. sorry, I had a brief moment where I'm like, wait, which direction does that go again?
Yeah.
So they'll mate, but it's not like at fantastical levels. So we tend to have to age the females and put more flies in a vial and put the cotton down a bit so that they're more crowded. So it's a harder cross to get to go than you would have for a wild type cross.
Right. So do you, in that context, like you mentioned how the hybrids that do mate with melanogaster are kind of like melanogaster. Do you think that that's because they want to mate with melanogaster, or because they're just receptive to mating?
Oh, like they're just sort of messed up and don't have a rejection response.
Yeah, basically, or like it just, if melanogaster are more receptive to mating, they're just less choosy for whatever reason. Like, because you can see that within melanogaster populations, there are some that are more and less choosy. Like in my context, right, the African France populations, the European ones are just like less choosy.
Yeah, so it could be. I mean, so these, so certainly, yeah. And what we were looking for was not finding out what loci or for melanogaster, hey, I want to mate. We're more interested in what are the loci that come from simulans that cause them to not want to mate with melanogaster males. So what is that female's willingness to mate? Is that being receptive? Is that being less choosy? I mean, that's that sort of nuance of that I don't know the answer to, and I don't even know how to answer that, how to parse that out in a way for a fly where you can't ask them what their motivation was in mating. So, but it's a fair question though, right? What exactly is the underpinning motivation of that mating? And we don't really know. They're not generally just willing to mate with any fly. So if you pair them with males that are really not great, then they don't want to mate. If you pair them with males who are from a more divergent species, they really don't want to mate. Even the simulans males, we pair them, they're not as excited about mating with them as with melanogaster. So they're not just inherently open to all the matings. They're still selective and having some levels of choosiness. But that level of selectiveness is much higher for simulans. So those females really don't like those melanogaster males.
Yeah, it just the context being, it might tell you different things about what genes might be important or what's at play, whether it's something discerning males or whether it's just like rejection behavior in general, or like what kind of cues that they're using to sense.
Yeah, so other works that we've done that did genetic mapping and identifying loci has led me to the conclusion that females are more often in a receptive state, and it's the absence of negative cues that's important. So they need to have some priming, so they need to have courtship, or else they're not going to be receptive. But in that, so they need to hear those cues, and if any one of them is wrong, then it's going to induce the rejection response. But it's not that they have to be primed with a perfect cue to be receptive, if that makes sense. And so why I'm saying this is that when we do genetic studies, we find that if we have even a single locus for rejection behavior, females will be rejecting. Whereas if we have a single locus for receptive behavior, they're still rejecting because all those other loci are still rejectionary behavior. So having a single permissive locus doesn't erase the rejection. So it seems that any presence of rejection is what it takes to have rejection. But to me, that implies then that the default underlying state is actually receptivity. And then you have all these potential cues. And if any of them say no, then that's what triggers that rejection response. And there's a lot of things that can trigger that rejection. So it's possible that threshold might just be lower for melanogaster, that they just don't, or higher, I mean, that they don't have to have as much of a strong of a rejection cue, or it could be there are fewer things that trigger that rejection response. So I'm not sure exactly what combines that to make that difference. And that's a great question. I think it's a question that I don't have a tractable answer to at this moment.
Yeah, that's interesting. Thanks.
Yeah, I think Erin is next.
Hello. I was wondering, I guess from an evolutionary perspective, and I don't know if you've thought about this or if people thought about it. So why would this selection or female preference allele arise in the first place, especially if they're preferring males that don't necessarily have a fitness advantage, and that the traits that they're preferring haven't evolved yet?
Well, it's not, I mean, I don't know if you can say the traits that they're preferring haven't, you mean within fruitless, or you're talking about the veiled preference paper?
The veiled preference paper.
Oh, okay. So yeah, the whole the whole theory of that is that there doesn't have to be a purpose. So it can just be by chance that you can have something that would cause you to have that preference that has no link to anything that actually is relevant at that moment. But there's no cost to it, because you'll still mate because your preferred male is not around. And then if something arises that matches that preference, then you'll go for it. In many cases, it might be that that preference is actually not great. Maybe you really like something that's a really poor fitness, and then that allele will be purged, right? That the moment that you have something that matches that, and then you're at a fitness disadvantage because you're mating with somebody who actually has poor fitness now, like it's not great. So the vast majority of the time these arise, there'll either not be a partner, or it'll be purged because it's not a great match in terms of fitness. But the moment you have something that links it to something that's a fitness advantage, then it'll spread through the population, and then you can get fixation of that combo. So I don't know if that answered your question, but essentially it's that why would it arise is that it would just be by chance, that it doesn't actually need. So often the models behind this needed a motivator, because otherwise there's a cost. And so what we're proposing is that there isn't a cost at first when these arise, that it can stay in there and the females will continue to mate normally, and this can just float around in the population without a cost.
I guess I was just curious, because it would seem like something that arose randomly would have to be like a point mutation, but it doesn't seem like something as singular as a point mutation could change something as complicated as like preference for a certain thing.
So what we find in, just going back to the work that I presented on today, so that fruitless locus, it's 2 nucleotides that are different in that P2 exon between melanogaster and simulants that cause an amino acid change. And actually one of them would be sufficient. So there's 2, but you could have a single one that causes that amino. Now, is that causal? That we haven't figured out yet. So we're in the process of testing that directly. But you can have single amino acid changes theoretically cause really huge differences in behavioral outputs. And we certainly know that single genes can have huge effects on behavioral outputs. So I don't think thinking that a single point mutation could cause a problem, like a shift in behavior like that is far-fetched. I think that's actually certainly very possible to have that happen, that we've got lots of evidence that would be a tractable possibility.
Thank you.
And Siwan, you have another question?
Yes.
Can I interrupt a little bit? So Dr. Moerhing have to go at 5 o'clock, so it could be the last question. All right.
Okay, Werner wants to ask a question before me again.
Or I can ask 2. If somebody else thinks of one, I'll answer 2.
Okay, great. So I'm actually curious about the fruitless. Can it be also serving as a kind of a preference allele, a preference gene to distinguish males that might have different mating success? I mean, can fruitless play a role in determining which males they like to mate with, or which males they less likely would mate with?
So I don't know if that's, so it's a great question. So we know that variation at P2 affects whether a female is receptive to melanogaster males or simulans males. So we see that difference based on that. But your question, I think, is getting at, does it affect variation within a species? So if you have variation, can it affect if a female is more receptive towards a male that has a slightly higher pitched song or a lower pitched song, for example, or in flies it's faster or slower pitched? And that we don't know. So we would need to do an assessment of if there's variation in that locus within a species, which we could do actually this reference panel that has sequence lines that we could look at that sequence. Again, what's challenging is at this point, I don't want to do that exploration until we know what's causal for variation in the behavior. So until we know what we should be looking for. So I imagine what we would find if we looked is a lot of variation in the regulatory region. So I know even among a few strains that we've looked at, we've seen variation in that regulatory region in the sequence. Does that matter for the female behavior? I mean, I don't know. We haven't linked that. So we certainly know there can be variation. Whether that variation is relevant for variation within a species, we don't know. And so one thing that we could do certainly is take these sequence lines. It's called the Drosophila Genomic Reference Panel, the DGRP, and we could assess them all for female receptivity. and then look at the sequence of that P2 and see if there's a relationship between receptivity and P2. So I would just need a grad student willing to embark on that to test that. So there's ways to get at that question and start to narrow down if that's causal or not. So we don't know is my short answer is we don't know. And then I gave a long answer of like how we could maybe try and get at that.
Look forward to the result. Thank you.
So someday I'll need to bring in a student who's willing to do that first.
Yeah.
All right. Let's say thank you again for the wonderful talk and taking the time for discussing with us.
Thank you all for your great questions. This is honestly, so my favorite part of this job truly is the mentoring aspect and the interacting with students and all of that. So I love that you have this after the seminar to give speakers a chance to interact with you. And I mean, students always ask so much more kind of gritty questions, like really they're not shy about asking like, okay, but what about this, in the way that like PIs are a little more hesitant about that. You know, they don't want to step on people's toes. And those questions are what make you think, right? So I think that's great. You guys came with some really great questions.
Thank you so much.
Thank you.
See you guys.